All right, I'd like to talk a couple of lectures here about epigenetics, one of the really important areas of genetics that's just booming as far as information content, and certainly for whatever your career is, and it has anything to do with genetics and association with genetics through medical or pharmacy or just whatever. Epigenetics is going to be more and more and more. In fact, even if you're not in genetics, just part of your understanding your daily life and how medicines and treatments are going to be developed. This is one where it's going to be a really important area. So I thought we'd talk a little bit about it. So let me share a screen here. And all right. So I just thought I would go through and some of this is just review. I know most of you have talked about, had some information about epigenetics, certainly just in general genetics, we talked about it, and it's been talked about molecular genetics and other areas. But I wanted to just kind of bring it all forward and let us think about what it means and what's the definition. Then we'll go through a few of the areas where the common ways that we see epigenetic modifications in genomic systems, and then we'll try to relate this to human systems, talk particularly about behavioral effects on humans and development. All right, so let's start out and talk about what is epigenetics? Okay, what is the definition? What is, well, I think most of us know that there's the genome, right? And then we're talking about epigenome, so it must be something that's related to the genome. And it is, of course. It's been realized that there's this area of, particularly from the regulation studies, from the science of understanding gene regulation, we've come to understand that there's this whole level of control above the genes. So genes are there, they store information, but you have to turn them on and turn them off and regulate their expression. And how that's done is through what's known as the epigenome, the layer above it. And these are tags that tend to turn genes on or off, sometimes permanently, sometimes short term. And it's a direct interaction, either directly with the DNA itself. Okay, so sometimes it's going to be an effect on the actual DNA code itself. Some other times it's on histones, as this illustration is down here. A lot of the activity comes from these areas where we've wrapped DNA around these complex of histones, and those bind the DNA. And if we want to get in to read the DNA structure that's in there, we have to open those up and make it available. Well, if you clamp it down, you can control it or turn it off. If you release it, you can allow it to be open. Okay. So the epigenome. Interestingly, 15 years ago, probably, if you read and you go back and read some of the literature in major publications, science publications, journal Science, Nature, the top tier journals, there were these arguments about whether epigenetics really was that important, particularly in evolution and expression, or whether no, it's all about gene mutation and sort of a Darwinian view of these changes. And it's become very clear over the last decade or so that epigenetics is really an area where there's massive control, and that control changes over time. And it's very, very important to what the final ultimate phenotype is going to be for them. So that's why we're going to talk about it a little bit. Okay. Well, we're going to make a point. Where do we see epigenetics in human systems? Well, one of the classic ways of studying any traits in humans are to study twins, particularly monozygotic twins, and MZs, monozygotic twins. And we can compare those to dizygotic twins to see what happens. And we can also compare monozygotic twins over time. And we know that they start out identical, right? You think about the fact that a monozygotic twin, by definition, the word means monozygote. So you start out as a single fertilized egg. That egg will then break into two, sometimes into three. You get triploid. You can get a triploids, but you can get three outcomes, but usually into two. Okay, that's the commonplace. Representing probably about 20% or so of what twins are. Okay, vast majority of twins are independent fertilization events, totally independent individuals. So those are dizygotic twins, monozygotic. But it turns out that monozygotic twins are not identical. Genetically, they start out identical, most certainly. and they tend to stay that way. But morphologically, it doesn't take long for them to start to diverge. Okay, now sometimes it's because they want to, they'll change their hairstyle or whatever. But phenotypically, we see differences as we start to age. I think most of you that if you have, any twins in your family, or if you know twins, as I did, I had good friends when I was young that were twins, and they were, but we could tell them apart. I mean, you'd say, "Yep, that's so and so." Nope, that's so. I could tell. We could tell them at a difference, distance. I mean, with pretty much no problem. Once you saw them, by that time they were 8 to 9, 10 years old. We play baseball. We'd say, okay, Gordon, you go out to left field, and Gary, you go out to the right field. And they which ones. Adults had a really hard time. They would look at them and wouldn't know who they were talking to. But so the point of that is that it doesn't take long for differences to start to appear. And certainly by older age, later age, we see real differences occur. And these are related to lots of different events that we're going to talk about. I know you've talked about some of that in general genetics about the aging process and the development. But what we're going to concentrate on is the epigenome and how the epigenome causes these differences. Okay, so kind of put this in the human perspective. Okay, there is monozygotic twinning. There's familial versions of that, okay, where there are families which have a lot of twins in them, okay. They just tend to have these combinations of twins that occur quite frequently. And in those families, and in fact, in all twinning situations, we end up with what are known very often as discordant phenotypes. Now, there can be discordant phenotypes during fetal development, okay, most certainly. That can be caused by intrauterine conditions, shared placenta, all sorts of things that can go on, okay? But as I was pointing out, as we go through time, we definitely start to see a separation in the phenotypic structures of twins. So we see this discordance grow more and more and more and more. And some of the ideas are that there are point mutations that occur because we have mutations that occur to our DNA throughout our life. They can be things like chromosomal mosaicism. Okay, so that your chromosomes aren't perfectly exchanged all around as the organisms develop, and that will interrupt. And that's a very mosaicism is a very large event that can lead to it. But you certainly can see discordance from that. Mitochondrial mutations. Don't forget, we've got this very important 16 kilobase plus unit of DNA that's located in the mitochondria, and you can have mutations there. You could have disjunctions between that in various ways, and certainly that can change. And they're very important. Energy production, ROS production, reactive oxidative species, and you get those. The more that you get, the more you get changes in your DNA. So you can, over time, you get all sorts of things going on. And we're starting to learn that there's it's been fairly well known for some time that X chromosome inactivation. So particularly, we're talking about females here with 2 X's. Through the Lion hypothesis, we say that it's random whichever one, the paternal or maternally derived X, is actually becomes inactivated, and that holds pretty much true. Except there are cases, particularly when there are cases when there's a chromosomal anomaly. That something that has to do with the X chromosome, you'll see sometimes favoring either the one that's got a problem, the chromosome with the mutation, or it'll favor the other one. You'll get a non-random patterning of that. And so even within twins sometimes, okay, female twins, the X chromosome pattern, who gets activated, if it's just a random process, you're going to get differences just from the fact that you've got those X chromosomes being spread out in different ways across the entire body. Okay. Now we're going to talk about epigenetics on this. So all those things are interesting, and we could certainly spend much of the semester talking about discordant phenotypes in twins. But I wanted, in this lecture at least, to talk about epigenetics. So we're going to talk about it as, okay, so what happens now when we start modifying that DNA, changing that DNA over time, and it doesn't change in one individual as it might in the other. Okay. I want to take a really quick little side trip and ask a question. Is this possible? Okay. So let's think about our pedigree symbols. We're looking at familial monozygotic twins over here, and every time we have a monozygotic twin, we get this little thing. And sure enough, you look at them, that's two females and two females and two females and two females, and it's always the same sex, right? We always, if we have it, we have two males or two females. And so is this possible? And just as a side note, absolutely it's possible. It's very, very rare, but it is possible to have monozygotic twins, technically, who have different sexes. And you say, well, how is the world is that possible? Well, it's possible from a number of things. One is that you may have an XY fertilization, and when you split, for whatever reason, the replication process starts fairly early on, right? Obviously, when you the early division of the zygote, and you can make a mistake. The Y chromosome doesn't have a lot of linking up to the X, and so sometimes it's just lost, and you end up with one side developing into male at normal XY, the other one losing a Y or misplacing it or having a duplicated X problem, and you end up with a female on that side. Another way that can happen that's been known, again, this is really, really rare, but I just wanted you to know it could happen since we're talking about advanced genetics here. The other way it can happen is that you have a situation like this where you have an X-bearing egg, an XY bearing sperm, something like that. And so you would be expecting to have a Klinefelter's, right? Klinefelter's XXY male. But in fact, what happens is you get this XXY and the system sees that. And on one side, after the split, it spits out the Y, okay, and you end up with a female. On the other side, it spits out the X and you end up with a male. And there's been some pretty good case evidence that things like this do happen. So is it possible? Yeah, it actually is possible to be identical but have different sexes. And it's like, so they're not truly identical, once again, at the end product, at the phenotypic level, but they're monozygotic, okay? All right. So let's go back and think about this. So just a quick review of epigenetic modifications. And the two biggies are things that have to do with methylation and chromatin remodeling. And then we'll talk a little bit about microRNAs, because those definitely have a human impact system. So how does epigenetic control happen? Well, remember you've got two places that you can have particularly methylation going on. One is on the DNA itself. Okay. The other is in this chromatin area. So that's the chromatin remodeling. So you have methylation that can occur on these histones or on the DNA around the histones themselves. Usually it's on the histones. And we have these little arms that kind of either fold in and tighten around the DNA to hold these loops on, or we can loosen it and let the DNA freeze so that it can be read. Okay. And so you have two major protein groups, enzyme groups that are involved. One falls under this group known as HAT, histone acetylators, okay? And then you've got HDAC, which is histone deacetylators. And these are ones that acetylate or deacetylate the system, turn them on. Then you have methylation, ones that will come along and do the same thing, okay? So we can acetylate or deacetylate these particularly these little pieces that hang off of histones, okay, the little arms that hang out. Can be acetylated or deacetylated. That's one of the big ways we can do it. Or of course we can, and we can have methylation as part of that acetylation process, right? Methylation is the action there. And generally these do things like they can turn on systems, okay? If you have HAT acetylators, that tends to often open up these histone arms and you end up with an open DNA where it can be reached and read, and the RNA pole can come in, all the transcription factors can play. Deacetylation tends to lock it down, and you get a switch off, you get a silencing. Genes, like genes that produce digestive enzymes in your hand, the cells in your hand have those genes, but you're not producing any there, skin cells. So what happened? Well, they've been permanently turned off, and one of the ways they're turned off is by this chromatin remodeling, shutting down whole regions of your chromosomes so that you don't have any expression of those genes. Right? And then of course you can have direct DNA effect, right? Where you can methylate the actual bases themselves. One example that's common is the cytosine modification, which can be methylated to have a methyl group added on to the number 5 carbon, and that becomes 5-methylcytosine. It fits in, it works, everything's fine. And in fact, when you do this, there's actually a ubiquitous enzyme known as DNA methyltransferase or methylase, which when does it. So one point of that is when you replicate and it sees that you've got a methylated one over here, in the new strand, it will actually methylate that one as well, where it's appropriate. And you can have multiple ways. This shows that you can have an unmethylated, you can have a hemimethylated or half methylated side. And then of course you can have a fully methylated where both parts of the system are methylated in forms. So it's just it's a way to control it, okay? So what's the end product of this? And again, I'm not going to get into all the methylation. That's chemistry and the mechanics of that. But methylation usually inhibits transcription of the genes. Okay, especially occurs when it's in the region of the promoter. And you may remember that there are these CG islands, they're called. Okay, they're CG repeats. They're found all over, and they're very commonly found in promoter regions. And it's those CG islands, and they can occur within genes as well, within the actual transcriptional areas, but they are generally up in the promoters, and they're just strung along. They can be used, from hundreds to thousands of these repeats, nucleotides long, and they're areas that tend to get methylated. Okay, so this leads to a couple of things. These are things like housekeeping genes. Okay, genes that you need in every cell that's going to help the mitochondria work, or transport genes, or P62, sequenceosomes, the HDAC genes, they're turned on constitutively. They generally have CG islands that are unmethylated, although they may be methylated at certain times. The tissue-specific genes, things that are turned on in your hand but not turned on in your some muscle or something in your brain, a neural cell that's turned on. These are very often regulated by these CG islands. So they're usually regulated by the methylation of those in combination with acetylation, methylation of the histone regions themselves. Okay, so this is just kind of a classic thing you see with transcriptional slice. And what it does is when you get a CG island up here, okay, and you methylate that island, so we've methylated it in a number of places, right? You just simply the transcriptional factors can't functionally see it as an aberrant form. It's not the signal they're perfectly looking for, and it tends to block it. So very often that's why it's in the promoter region, somewhere this is showing around the enhancer, but wherever in the promoter region. And actually, if you go and you go to GenBank and you pull down the promoter region of lots of genes, sure enough, particularly those that are have strong control, you'll often see 1 or 2 or 3 even CG islands as part of that. Okay. So you've got this going on. So what are some of the factors that affect the epigenome? Well, one of the big ones and the ones that we talk about is things like behavioral patterns. Exercise, people that exercise versus people that don't, that are sedentary, have different expression patterns of methylation and epigenetic forms. Stress, people that stress tends to do it. So how do we know this? Because they're fairly simple, and we're actually going to look at some on the next lecture. But I say simple. They're not difficult to do, but they do take a bit of technical skill. You can look at either methylation of single gene regions. Okay, we can do that fairly easily, or you can look at whole genome methylation levels. And so there've been all these studies been done about looking at using methylation as a primary form of looking at whether someone who's under stress or not under stress, how much of the system is methylated, how much is not methylated, same thing. And then a big one is diet. Okay, methyl groups. Where do we get the what are things that come in with methyl groups are through our diet. And so your mom was right about you should eat your veggies. Because you get a lot of methyl groups there, you get a lot of things that protect you, a lot of things that turn on. If you're eating a lot of processed food, you get a lot of times the bad side of all that, okay? And how do we know that? Well, we know that because food provides major nutritions and nutrients that are required, such as folic acid and the B complex vitamins. These work in, as methyl donating nutrients, and they keep the system functioning. They help us regulate as we need to regulate gene expression. Okay. And so remember that, you got methylation can work both ways. It doesn't always turn things off. It can turn things on and upregulate and downregulate as well. How do we know as well? This is a classic study, and you'll see it all over the place. But it's been well worked out. As a really good example of this came along, we're looking at a Goody gene. So Goody is a color in mice, right? So a Goody is this color, that kind of the gray mouse that you would see running around your house, house mouse, okay? The Goody gene is turned on for a fairly short time during development, okay? The mRNA is only made for a fairly short time during fetal development. And actually right at molting, because when mice are born, they're a certain hair, and then they lose all that and they molt for adult pelage color. The Goody genes are active, but then they're turned off. Okay, they're turned off by methylation. Okay, methylation in a control region. Right. If you do this, you get a normal mouse, right? Okay. Well, there have been these mice that have been modified genetically so that they don't methylate. Okay, there's the CG islands have been disrupted. There's other things. There's some naturally occurring variants that turned out, but there's some deletions where they've deleted out some of the control region. So basically, what they've done is made it impossible to turn off that gene. So the agouti gene tends to be active throughout life. Okay. Well, what you get is the difference here. You get a normal one over here, or you get this sort of obese mouse over here, and they truly are obese. In fact, by 6 months or so of age, they're so big, they're like 2 or 3 times the size of a normal mouse as far as just fat composition. They have lots of cancer forms, diabetes. They're good models for studying diabetes because they will do that. They get all sorts of problems. Okay. Well, epigenetically, okay. So what? Well, it turns out that if you take one of these obese mice, okay, these agouti expressing mice, and you take them and you supplement them with methyl groups. Okay, so you give them food that's rich in methyl groups, right? When they will make mostly good, healthy offspring. Okay, most of their offspring will be healthy. If you don't, you just leave them alone. Don't give them supplements of any extra food. Let them feed that they want. Most of their progeny will be unhealthy. They'll eventually be obese mice. You'll get a few, of course, that will not. But this basic argument is that adding methyl groups, adding the nutrients needed to do the methylation, okay, will shut down this gene itself. Okay. Now you say, well, how can it shut down if you've cut the gene out? Well, you don't. Okay. I mean, they do the experiment with it cut out so that you can see that you basically get this outcome. But the other variations where they've just gone in and blocked the methyl part of it, then this is where you get the difference here. Okay. So you have a control where you can force it through. You have a regular control where everybody's normal and goes through, and you have this control where you completely blocked it off and you can't methylate it no matter what you do. And then you have ones that are, aren't methylated but could be, and this is the result that you get from that. Okay? Now, what's really, really interesting about this is that if you take these mice right here, okay, take one of these guys that's come from this obese mother, okay, so genetically it's got what its mother's got, and you don't supplement it, you don't give it any methylation groups, it will produce normal offspring. Okay, this is arguing that this modification, this protection lasts multiple generations. Okay, well, this is what's known as epigenetic memory. We're seeing more and more cases of this, and in humans, where this plays a big role is things like when you have females who are drug dependent. Alcohol can do the same thing. Cigarettes can do the same thing. Just exposure to work environments, things can do it. But one of the examples would be a meth baby. Okay, so mother's on meth. Babies often have problems anyway. But what this is saying is that it's not just that baby that's going to be affected, but it's the grandchildren that may as well be affected as well. This memory can work both ways, right? It can be a good thing like this, where you can straighten it out and get it moving toward being back to normal. But there's also evidence that you could have just as much detrimental effect on it. So eating really bad foods, being obese, being all sorts of things that will lead to children who have a similar pattern, but it also can lead to grandchildren who have a similar pattern. Conversely, healthy diet, exercise, doing things properly to correct what your mother gave you or your dad gave you can often may be able to pass on to your grandchildren. They may come from it well. So it's an interesting problem, and hasn't been highly discovered yet in humans, although there is good ideas and certainly some good examples where we think epigenetic memory is going on in humans as well. And one of the places that works out is in Sweden. Sweden has an amazing system for keeping track of everybody's health. Their DNA, we talked about before, their DNA is looked at. Their health records are all maintained. They've looked, and Swedish people have this system. Well, back in World War II, okay, they were isolated. Okay? Germany had occupied it basically, but they had just cut it off, and people were just starving to death. There's a massive, huge shortage. Okay, well, by the time they got to be, the grandchildren came along, this food shortage actually was related to things like an increased lifespan. Okay, so not just the children, but the grandchildren of these individuals who were basically in a starvation situation appears to be that they had an increased lifespan. Those individuals, when they had plentiful food, came back, have a decreased lifespan due to heart disease and diabetes and things like that, which obviously are not just purely epigenetic, but suggest that there are cases where this memory and where nutrition can play an important role. Okay. So the question here is, in these studies, is it really epigenetic? Okay. All right. The last of these that I want to talk about in this lecture are some microRNA modifications. Okay. And the example that I will use here is in schizophrenia. There is in schizophrenia various forms of it. About 2% or so of what right now is the estimate of individuals who become schizophrenic show up because of a mutational problem that's related to microRNA production. Okay, this microRNA production has to do with microRNAs are these where DNA is read, it's transcribed. That's not going to become a protein-producing RNA. It's going to be cleaved. Most of that and then occurs out into the cytoplasm, of course. And if you have an overexpression or an underexpression of these microRNAs, you can end up with either too much protein produced, as we'll see in a moment, or not enough protein produced. Okay, loss of function goes on because the microRNAs interact with normal messenger RNA in the cytoplasm, primarily cytoplasm, to cause, to break down the message, or to, in some cases, to activate the message. So how does this work in schizophrenia? Well, we're not sure, okay, exactly, or that say scientists aren't sure. There's been a lot of speculation, but there is a good mouse model that does show that in a relationship here. Now, again, I'm not talking about all schizophrenia. Okay? I'm talking about a few cases, some proportion of schizophrenia, and a propensity for those individuals who have certain known mutations to end up with behavioral problems as well as eventually, ultimately, potentially schizophrenic behaviors. Where this occurs is in a microdeletion. Microdeletions, remember, somewhere usually about a kilobase or maybe 10 or 20, 30 kilobases long. It's on chromosome 22, okay, so one of our smaller chromosomes, in the Q11.2 region, so right here. In mice, chromosome 16 has some very similar genes, okay, and they're known to be on 16. So there's been some deletion studies done So make these mouse models where they are deleted on 16 for the same region that's equivalent to this microdeletion in 22 in humans. When you look at those mice, they have, and this is again a heterozygous condition, but you could do it whatever. In homozygous condition, they basically don't function. It turns out that what's located there, both on humans in 22 and mice on 16, are these genes that are critical for early development, okay, embryonic development of the neural system, okay? And the way it works is through mRNAs, through these microRNAs, okay? And the genes that are affected here are the GGCR8 gene, which is downregulated, okay, it's cut down, right? And it's responsible for modifying and cleaving and actually some other things, turning some things on, of about 10, 20% of all known mRNAs. So what it basically is, you downregulate this gene because it's not there, okay, it's been deleted, and it basically stops these mRNAs from forming, okay? In the same region, there's the MIR185 gene. It's also downregulated, and it has a definite effects on some other, it's not going to make an mRNA, but some Golgi-related genes and other things go on, okay? Both of these, however, target, ultimately target, particularly the microRNAs, the MIR22 gene. This is the gene that's involved in neural development, okay? One of the primary genes involved in neural development, okay? And if you look what happens here, just look at the black line. This gene, this is the level, relative level of this gene activation. Embryonic and early neonatal times, there's a high activity. And as we reach adulthood, it basically turns itself off, okay? By the time we reach certainly parturition, it's very low in its expression. By the time you reach adolescence, it's turned off. You don't use it anymore. Okay, our neural systems have pretty much developed. In the deficiency in the mice, okay, what you see is that this gene stays active. Okay, it stays at a fairly high level. It's the highest again at embryonic, but it never really drops down to anywhere close to a wild type level. It stays active. It's upregulated. Why is it upregulated? Because the mRNA and microRNA, part of it is that the microRNAs aren't there to break the message apart. So even though the message is being transcribed, it gets to be making a full protein. So you get more of it. Well, what does it do? Well, it does a lot of things, okay? But in neural system development, there's this pruning and other activity that we'll talk about later. But one of the things that you see with these in the wild type is that you get in a dendrites, you get the dendrites forming and all, they look proper and forms, okay? So you get a neural cell that looks right. In these deficiency mice, and it correlates with what we see in the brains of young children who have these microdeletions, is that you don't get near the dendritic formation. Dendritic structure is not there. You get this less bushy form, you don't get the connections, etc. Right? And as this continues to go over time, it tends to be more and more. So while what we see is that they're in these children that have these microdeletions, while the cortex is as dense as it should be, the connections, particularly in the hippocampus and other areas, are not the same. So it ends up with this problem. So what happened here? Well, it didn't have anything to do with this gene per se, right? There's no mutation in that gene. The control regions, the epigenetic control of that is up here. Now, why is this epigenetic? Well, it's because it's a microRNA example of how this occurs. And so it's thought that while this is the way it works in the specific 1-2% of his, these cases where we have schizophrenia, the questions now are, what's controlling this region? Is if we put this region back and look at a normal situation where there's no deletion, is this same type of effect due to epigenetic inputs, right? Because one of the things that goes on is that schizophrenia generally, there's might be behavioral issues and things, but you don't really see schizophrenia until early adulthood, late adolescence, somewhere in that the teens and early 20s is when it really kicks in. So what's that 15-20 year period? What's going on? Well, it may be that you've got some epigenetic influence. You've got some control situations going on that affect. So it's right now, it's an open area, but it's certainly one of interest trying to figure out how these microRNAs and their locations and the control of those microRNAs themselves plays a role in things like schizophrenia. All right, the next lecture series, we're going to take epigenetics and move more into behavioral effects. We'll actually come back to schizophrenia and talk some more about that, but we'll also talk about a whole suite of other behavioral systems that are involved.